Yes, everyone. Hi. I think so that I am live and I am clearly audible and clearly visible to each and every one of you. Yes or no? Let me know in the comment section if it is yes. Let me know in the comment section, guys, if everything is going well. I am live, I am visible, I am audible. Great. Hi, Shivesh. How are you? Okay, chalo. That's pretty good. That's pretty good. So since we have completed few of the chapters in Hi Bala, how are you? So we have completed few of the chapters previously, right? Genetics principle of inheritance we have completed. So today I have brought you another chapter which is the molecular basis of inheritance, okay? And this chapter, you feel like this chapter is kind of hard because basics are not clear, maybe, or maybe the concepts you are not able to gain properly, right? So today is a very basic class. I'll tell you what things you have to remember in this chapter and how we are going to proceed with this chapter, okay? And I'll make sure that you, after completing the whole series, you will feel like this chapter, this video, this classes was worth it. Okay, everyone? So should we start? Start karu? Great. Okay. So before that, I would like to start with a very basic thing. I'll consider that you don't know anything, okay? But it's not like that. You people know. But as a teacher, I'll consider you don't know anything, okay? So just remember, let's talk about a cell. Let us talk about a cell. What is cell, sir? A cell is basically the living and the structural unit of life, functional and the structural unit of life. Yes or no? Your hand is made up of cells, hair is made up of cells, everything is made up of cell, or I should say each and every living organism is also made up of cell. Yes or no? Yes, very much clear. Now if I go to the structure of the cell, people, so see what is going to happen. Here we have the cell membrane. Yes or no? Ignore my drawings, guys. Ignore my drawing, okay? So here we have the cell membrane, and inside the cell membrane we have the nucleus. Yes or no? And then inside the nucleus, what do we have? So we have some chromatin material which is known as DNA or chromosomes or something like that. Yes or no? Yes. These are the chromatin material. Or if you right now, if you are confused with chromatin material, chromosome, let's consider it as the genetic material. What they are? They are genetic material. Yes or no? Yes. So basically each and every cell inside our body is having this structure. सेल है, न्यूक्लियस है, न्यूक्लियस के अंदर क्या है? न्यूक्लियस के अंदर है हमारा जेनेटिक मटेरियल. यस और नो? इफ आई आस्क यू शिवेश एंड बाला, व्हाट डू यू हैव इनसाइड योरसेल्फ एज अ जेनेटिक मटेरियल? डीएनए और आरएनए. क्या है तुम्हारे पास? इज इट डीएनए और आरएनए? व्हाट डू यू हैव, गाइस? Bala and Shivesh, I want the answers. Yes, obviously, Shivesh, we have DNA inside us, right? We have DNA inside us. So how things work? Things works like DNA is going to give order. DNA is going to give an order, okay? DNA order dega and DNA will ask that to make some RNA, right? DNA से क्या बनेगा? RNA. DNA will make some RNA, and this RNA, yes, exactly, this RNA will make some proteins. Yes or no? This RNA will make some protein, and let's suppose that this DNA was containing the information of your eye color, कि तुम्हारा आंख कैसा होगा, पीला होगा, काला होगा, गोरा होगा या ब्लू होगा, whatever, right? So this DNA was containing that information. अब वो information RNA के पास गया और RNA ने protein बनाया, and from the help of this protein your eye color is decided. Yes or no? This much is clear? Yes. So in this chapter we all are going to see that how this is happening, how DNA to RNA is being made, RNA to protein is made, how protein is being expressed, what is the structure of DNA, what is the structure of RNA, from what structure the DNA is made up of. Everything, guys, everything we are going to deal in this chapter, right? Okay, I'll carry on with the English, don't worry. Little bit na flow mein I just speak English, Hindi, but don't worry, this is a full English channel. This is a full English channel, guys. Thank you. Bala, from next time if I speak English now, you just comment, sir, what is this behavior? Okay, chalo, let me start again, okay? Chalo. Now see, there was a person called the F measure. What was the name of this person? F measure. Yeah, just try to remember right now only, okay? We will clear it out everything here only. There was a name of the person which was F measure. I'll repeat so much time now that you will be able to remember this. So what happened to F measure? F measure was going somewhere, okay? Measure was going somewhere. He was like, I'll go to the park, I'll play and all those things. But what happened? F measure got hurt. F measure got hurt in his hand, and after few days infection happened in his hand and pus got created. What happened? Pus got created. Okay, do you understand pus? When infection happens, so pus gets created, yes or no? But F measure was a scientist. F measure was a scientist. He did what? He took out that pus and put it under the microscope, and he saw some cell, and inside the cell he discovered nucleic acid. He discovered what? Nucleic acid. Remember? So F measure was a person who discovered nucleic acid. F measure was a person who discovered what? Who discovered the nucleic acid. But remember, this was a person who didn't give the term. He said that it's not nucleic acid, it's nuclein. F measure said that it's nuclein. F measure said that it's nuclein. So there is one more person who came that F measure, bro, it's not nuclein. Let's give it a term, says nucleic acid. So Altman was a person who gave this term. Okay, again I am repeating. F measure was what? F measure was what? F measure was a person who went to a park, got hurt, that wound got infected, pus got created, and he took out that pus, saw under the microscope and discovered the nucleic acid. But he didn't say that the term nucleic acid. He said that I discovered nuclein. There was another person, Altman, who suggested the name nucleic acid. Bala and Shivesh, is it clear to everyone? Is it clear to everyone? Is it now okay? You won't forget? You won't forget? Clear? Okay, chalo. Now, let's move on. Now, there is one more question, guys. DNA is so big. Yes or no? DNA is so big, right? Nucleic acids example are what? DNA and RNA, and they are so big. So, from which material they are made up of? So, see here, these nucleic acids are made up of nucleotides. These nucleic acids are made up of nucleotides. Thank you so much, Bala. Thanks a lot. This nucleic acids are, thoda sa main smile kar lu. Should I smile? Okay, chalo. Now, this nucleic acid is made up of nucleotides. What is this nucleotides, sir? See, if I ask you a question, guys, protein is made up of what? Then so you will be like, sir, this so I know. Protein is made up of amino acid. Yes or no? Protein is made up of lots and lots of amino acids. So in the similar manner, guys, this nucleic acid is made up of lots and lots of nucleotides. So to understand the structure of DNA or RNA, we need to understand what is nucleotides. Yes or no? Yes or no? Let me know. Chalo. Great. There are students which are absent, I know. I'll give the punishment to them. Yes or no? Shivesh? We should give punishment to those students. Right? Chalo. Let's move on. Bala, in case of any doubts, let me know in the comment section. Okay? Whatever the doubts you have, a small doubt or a big doubt is a doubt. So let me know in the comment section. If you want me to repeat anything, comment. Okay? Chalo. So now let's talk about the nucleotides. Exactly. These nucleotides are made up of what? These nucleotides are made up of nitrogen base, and what? Along with, there is what? There is pentose sugar, and what we have? Phosphate. Yes or no? Yes. We have phosphate. Okay, this is nucleotide. So nucleotides are made up of three things: nitrogen base, pentose, sugar, and phosphate. But guys, if I talk about what do you understand by nucleoside? Nucleoside. So see, phosphate is what? Phosphate is a very arrogant person. Phosphate is what? It's a very arrogant person. So this nitrogen base and pentose sugar says phosphate, go to side. Go to side. Phosphate is a very arrogant person, and this nitrogen base and pentose sugar says to phosphate, phosphate, go to side. So nucleoside is what? Nitrogen base plus pentose sugar. Nucleoside is what? Nitrogen base to, yes, nitrogen base plus phosphate sugar. Sorry, what I'm saying, nitrogen base plus pentose sugar. Yes or no? Okay. Now one by one, what we are going to do is that we are going to learn the structure of these nitrogen base, and I promise you that I'll make you learn. Okay, I'll make you learn all the structure. So first of all, let's make the uracil. Okay, there are nitrogen bases. How many types of nitrogen bases? First of all, let's discuss that. Yes, anyone in the comment section, guys? How many types of nitrogen bases? How many types of nitrogen bases? Two types, sir. Basically, pyrimidines and purines. How many types? Pyrimidines and purines. Yes or no? Pyrimidines, just keep an eye on me so that I won't make spelling mistakes. Pyrimidines and purines. Four types. Okay, majorly two types, and then after that we will divide it. Yes or no? Yes. Yes. Purines. Now see, what happens na? Naam bada, like name is very big name. Kaam, work is very small. Name very big, work is very small. Like sometimes happens na, when you see an Insta reel and you go to that restaurant or a cafe and you will be like, this is what? So it was what? It was shown very hi-fi, and when you went, it was very small. Okay, so pyrimidines is same. It is what? The name is very big, but the ring is small. The name is very big, but the ring is small. Single ringed structure. Pyrimidines are what? Pyrimidines are single ringed structure. Pyrimidines are single ring structure. Okay, naam bada, kaam chota. Name is big, work is small. Now, purines is what? Purines is name small, work big. So, they are made up of two rings. They are made up of two rings. Yes or no? Two rings. Right? Example of pyrimidines. Example of pyrimidines is cytosine, uracil, and thymine. Example of pyrimidines is cytosine, uracil, and thymine. You give CUET exam. Yes or no? You give CUET exam, that is pyrimidines. Cut, okay? CUET exam, cut pyrimidines. And purines are what? Purines are AG. Purines are what? AG. Purines are what? AG. Like there was a wife. She said that I won't make tomato puree. I won't make tomato puree. So she said to his husband that, he's not her husband, she said to her husband that, AG, bring purines. AG, bring purines. Clear to everyone? Clear to everyone? Any doubts? You won't forget which is purines and which is pyrimidines? Okay? Chalo. Great. 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 Should I move forward? Shivesh is laughing. Chalo. Let's now discuss the structures. Let's now discuss the structures. Shivesh, you will be also that kind of husband there. Don't worry. We have to do lots and lots of work. I really don't like to do one thing. Like, I'm not married yet, but my mom always says me to bring vegetables, and that's the worst thing I can do. Like, I really go there and I am literally get confused that what vegetable I should bring. And when my mom and dad will go na, I'll say them, bring something good. And the similar thing which I have to do is that then I also don't understand what should I bring. So vegetable thing is very bad for me. Chalo, let's get back to cytosine. Okay, cytosine, what you have to do is that you have to make a benzene ring kind of structure. What you have to make? A benzene ring kind of structure. First of all, let's make this. Let us make a benzene ring kind of structure. Okay, but here what? Here what? You just give the double bond at this position and this position. Okay, benzene ring kind of structure and give the double bond at this position and this position. ऊपर में what you have to do is that on top of that you have to put nitrogen. On top of that you have to put NH2. Okay, then write here N also, and then double bond O and H. Okay, what you have to do is that benzene ring structure, make a benzene ring structure. Okay, in top of that, top of the benzene ring N, and in here also N, then O. No, notch. Okay, how you have to remember it? Cytosine notch, cytosine notch. Okay, cytosine notch. How notch? NNOH. How notch? NNOH. Clear? NNOH, cytosine notch. Okay, NH2, N, double bond O, H. Clear? I think so. This will, you will be able to remember this, right? Now uracil is what? In uracil, what you have to do is that just you have to remove what? Just you have to remove the NH2. Just you have to remove the NH2. Here in uracil, everything will be same. Everything will be same. Instead of NH2, we will have O. Instead of this, we will have O. Instead of this, we will have O. Okay. One more thing I have forgotten. I have not written N here. I have not written N here. I'll write N. Okay. Clear? Which nitrogen is more basic? I'm not your chemistry teacher, anonymous. I think so. This answer should be answered by the chemistry teacher. I think so. I should not answer this, right? If I'll answer wrong, then you will be like, oh, sir, you don't know chemistry. Huh? I think so. I think so. Basic, I really don't know. So you have to figure it out with your chemistry teacher. Okay. Sorry for this. Cytosine. Cytosine. Okay. This was cytosine. And this is what? This is the uracil. This is the uracil. Don't worry. We have a surprise for you. Jaldi, you will be getting a chemistry teacher. Don't worry. So how cytosine and uracil is different? Cytosine notch और uracil में N हटा के O लगा दो. Uracil में N हटा के O लगा दो. Okay. Then we have thymine. Thymine तो is very easy. Thymine तो is very easy. Make uracil. Okay. To the structure of thymine, you make the uracil. Just copy uracil. Just copy uracil. And what to do? And what to do? Just add CH3 here. Just add CH3 here. Clear? This is thymine. This is thymine. Clear to everyone? Yes or no? अरे याद कर लो. I am making you learn so easy. Cytosine notch. Uracil N को हटा के O. Thymine. Copy of uracil plus CH3. Copy of uracil plus CH3. Is it clear? Is it clear? Is it clear? Yes or no? Chalo, great. Shivesh says that I am amazing. Thank you, Shivesh. Thank you, Shivesh. Padh lo bhai, study also. Study also. Otherwise, when you will not score good in NEET, then you will say, sir, you were not amazing. Yes? So you have to study. Saath saath mein padhte bhi chalo. Right? Okay. Now, let's move on and let's talk about the purines. Purines was what? Are you Anubhav Singh's Bassi's brother? No. My friend says that you are ruining the name of being Bassi, because when I crack jokes, they don't laugh. Unfortunately, guys. Chalo, let's get back to purines. So purines was what? There was the wife who is saying to her husband, AG, bring purees. AG, bring purees. Yes or no? So this AG is what? AG is guanine. So it is made up of two rings. One is pyrimidine ring and the other one is imidazole ring. Okay, one is pyrimidine ring and the other one is imidazole ring. So let me make the pyrimidine ring. In biology, see, basically, what's your status? Like, are you a very person who doesn't know biology, or you are pro at biology? So if you are pro at biology, 2 to 3 hours per day is also very fine. Yes or no? Yes or no? Clear? Is it okay? If you are pro in biology, then you should give more time. Clear? Chalo. Now we have what? Now we have NH2 here, then ring here, okay? And then we have N here, okay? Then we have N here, right? Okay? So 3N. How many N, guys? How many N, guys? 3N. How many N, guys? 3N. And then you make an imidazole ring. And then you make an imidazole ring. Yes or no? And then you make an imidazole ring. Clear? Clear to everyone? This is the structure of adenine. This is the structure of adenine. Naming kaise karna? How you have to name them? How you have to name them? See here. From, you start with this one. 1, 2, 3, then 4, 5, 6, then 7, 8, 9. Okay? 5 और 4 के बीच में डबल बॉन्ड होगा एंड इसके बीच में डबल बॉन्ड होगा और 2 और 3 के बीच में डबल बॉन्ड होगा. क्लियर टू एवरीवन? क्लियर टू एवरीवन? यस और नो? यस और नो? यस और नो? क्लियर टू एवरीवन? चलो, ग्रेट ग्रेट. Is it any doubts? Do you have any doubts, guys? Do you have any doubts? Chalo. Yes. So we have this adenine. Now what? Now let's have the structure of guanine. Structure of guanine. Let's talk about the structure of guanine. 1 second, बच्चों. 1 minute. Chalo. Structure of guanine. What's the structure of guanine? Let's see here. Again, imidazole ring कौन सा था? This is the imidazole ring. This is the imidazole ring, and this is what? This is the pyrimidine ring. Okay, this one is the pyrimidine ring, and this one is the imidazole ring. Any doubts? Great. Now let's move on with the guanine. Let's now move with the guanine. So guanine is basically what? Again, that imidazole ring will be same for everyone. So here what you have to do is that you make N double bond O, and then here we have, and then here we have again NH2, then N, and this one. My drawings are very good. Yes or no? So you have to manage it. Chalo. This is what? This is the guanine. This is the guanine. Clear? Naming कैसे करेंगे? Similarly, from here we will start 1. From here we will start 1. Okay? Clear? These are the structures which you have to remember. See, in NEET now, no one is going to ask you the structure, to draw the structure. Yes or no? This is the very clear. In NEET, you don't have to draw the structure. So what you should do is that you should take some points here. What points? See, in adenine we have NH2 here, and in guanine we have double bond O. Just remember this. In adenine we have NH2 here, and in guanine we have double bond O. When the option is going to come, you will be able to easily recognize it. Clear? So pura structure, you don't have to worry about the whole structure. You just pinpoint the difference and learn the structure. Nobody is going to ask you in NEET to make the structure. Yeah, in boards it's necessary, you have to learn the structure. So if you are giving boards, please learn it. Okay, now let's talk about the pentose sugar, guys. Let's talk about the pentose sugar. Now what does this pent means? Pentose sugar, what do you mean by this? You know, pent, 5, hexane, pentane, like this. So pentose means what? 5. So 5 carbon ring is pentose sugar, yes or no? Yes. So see how to make this. On the middle you have oxygen. On the middle you have what? You have oxygen. And now make the 5 carbon ring. Now make the 1, 2, 3, 4, 5 carbon ring. Okay. Make the 5 carbon ring. Clear? Yes, exactly. Now what, sir? See, now what? We if I talk about your hand, this is palm, and if I talk about your ankle, okay, so which one is bigger? Ankle is bigger or your your palm is bigger? Your ankle is bigger or your palm is bigger? Obviously your ankle is bigger, right? So to stand, to wear shoes, you wear in ankle, so ankle should be bigger. So here niche, in down part, be given preference to IUPAC. Yes, yes, yes. मैंने गलत किया क्या? मैंने गलत किया क्या? Have I made a mistake? No, guys. सही है. It's okay. Manjita, it's fine. This is correct. Check it once. If it's wrong, but it's not wrong, I'll know little bit chemistry also. Check it once. In biology, to guys, we do the numbering this only. In chemistry, I don't know. So let's get back to pentose sugar. So in pentose sugar, what you need? In down part, like in your feet, you wear what? You wear shoes, which is long, broad. So OH will be down. OH will be down. Okay. Niche wale mein OH will be down. See here. Here also OH will be down. But in this one, what's going to happen? In this one, OH will be up. In this one, OH will be up. So while you are wearing the shoes, OH will be down. Niche wale hisse mein, during the down part, OH will be down. Shoes pehenna hai hame, bada lana hai, right? So OH will be down. Here what's happening? Here H is here and the fifth carbon is here. CH2OH. CH2OH. Clear to everyone? Will you be able to remember the pentose sugar structure? Will you be able to remember the pentose sugar structure? Manjeet, Bala, Shivesh. Yes or no? Manjeet, Bala, Shivesh, will you be able to remember the structure? Tell me if you have any doubts. Tell me. Manjeet, is it clear? Manjeet or Manjita? Manjita. Clear? Okay. Easily remember ho jayega. Oxygen 5 carbon you make. Okay. In this carbon 2 and 3 carbon. Juta pehna to wear them shoes. Shoes is big. OH. Clear? Like fructose, exactly. And now what you have to do is that you have to make deoxyribose sugar. What you have to make? Deoxyribose sugar. This is what this is. Ribose sugar, which will be present where, sir? It will be present in the RNA. It will be present in the RNA. Yes or no? Yes. So if I have to make the deoxyribose sugar, so what? Steal this oxygen. Steal this oxygen. Remove this oxygen. Then it will become what? It will become your deoxyribose sugar. It will become your deoxyribose sugar. Yes or no? Yes or no? Clear? Why ribose forms furan ring and not pyranose ring structure? Manjeet, I am a biology educator. So let's focus on biology. Thoda sa bio. I'll ask the physics educator, sorry, not physics, chemistry educator to help you with these doubts, okay? Yes or no? Okay, Manjita? Right? These are the parts of organic chemistry. Why they don't form the furan ring and pyranose ring structure. I think so chemistry educator will be the right person to explain this thing. Yes or no? Okay. Manjita, clear? Chalo. Now, whatever doubt you have in biology, you ask me. Clear? Clear? Okay. I am studying right now. I'm teaching you this one. Molecular basis of inheritance. I'm teaching you the molecular basis of inheritance. Okay. Chalo. Right here. Any doubts? Any doubts here? Any doubts here? Pentose sugar, is it clear? This is what? When we remove the oxygen, it became deoxyribose sugar. When we remove the oxygen, it becomes deoxyribose sugar. Clear to everyone? Yes or no? Okay, chalo. Great. Now let's have phosphate structure. Let's have phosphate structure. What is the structure of phosphate? You all know. This to you all know. Yes or no? Everyone knows. POH, double bond O, double bond OH, sorry, OH, OH. So what you have to do is that P is there, all the three ends is OH, 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 and on the top of that you have double bond O. All the ends are OH, 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 and top of that you have double bond O. Yes or no? Is it clear? Is it clear? Now, guys, phosphate is acidic or basic? Yes, phosphate is acidic or basic? Manjita, Shivesh, Bala, phosphate is acidic or basic? Phosphate is acidic or basic? I'll teach you this chapter so well na, that you will feel when you won't have any doubts in translation, transcription, anything. Acidic, exactly. Phosphate is what? Phosphate is acidic. Yes or no? Yes. And it's a negatively charged structure. And what? And it's a negatively charged structure. Negatively, yes, exactly. Negatively charged structure. It's what? It's negatively charged structure. Okay. Now see, we saw what? We saw the nitrogen bases, sir. Yes. We saw the ribose sugar and deoxyribose sugar, and we saw the phosphate. Now what we are going to do is that we are going to combine them all and make the nucleic acid. Yes or no? We are going to combine them all and make the nucleic acid. Clear? So see here. What happens here? If I talk about the DNA or RNA, so the backbone is made up of phosphate. The backbone is made up of phosphate, and here what do we have? Here we have the nitrogen bases. Here we have the nitrogen bases, and again here we have the phosphate, and again here we have the phosphate. So this is the basic structure. This is the basic structure. Yes or no? This is the basic structure. Clear? Okay, now let's make a proper structure of this. Let's make a proper structure of this. So we have phosphate. Yes or no? Let's make the phosphate. P double bond O, P double bond O, this O, O negative, and here we have O negative. Yes or no? Yes. Then this O is going to join where? This O is going to join with the pentose sugar. Which carbon? 5 prime carbon. Yes or no? See here, CH2 hydrogen, this oxygen, and we have the pentose sugar here, H, OH, H, OH, and this OH will be up. Yes or no? Let's suppose here we have the OH and H. Yes or no? Clear? This is what? This is your phosphate and this is your sugar. This is your phosphate and this is your sugar. And what is this? Nucleoside or nucleotides? What we have made right now. Is it nucleoside or nucleotide? Is it nucleoside or nucleotide? Phosphate plus sugar. It's nothing, guys. It's nothing. Sugar and nitrogen base is nucleoside. Yes or no? And all three are known as nucleotides. Clear? Okay. Now I'll make N-glycosidic linkage. Now I will make N-glycosidic linkage. See, this H, tide, Bala, tide. Why it's joined only to the 5th carbon? See guys, here we have the 5th carbon, right? Here, it will not join to the 4th carbon. It can't join. See, there is no group which can be removed here. Yes or no? In this carbon also. In this carbon also, no. So attacking 5th carbon is very much easy and it can remove this group. Which group? This group. This OH can be removed very easily. Yes or no? Yes or no, Manjita? That's what the reason why it's joining to the 5th carbon. थोड़ी बहुत chemistry आती है मुझे. Okay? चलो. Now what happened? We are going to form an N-glycosidic linkage. What we are going to form? N-glycosidic linkage. So the 9th carbon, sorry, the 9th nitrogen in this nitrogen base, the 9th, yes, exactly, the 9th carbon, the 9th hydrogen in this nitrogen base, what's happening? It is going to react with this OH, it is going to react with this OH, and they are going to form a glycosidic bond, and they're going to form a glycosidic bond. Yes or no? Yes or no? Let me know in the comment section. And they're going to form a glycosidic bond. Clear? What is this glycosidic bond, guys? What is this glycosidic bond? What is this glycosidic bond? Remember in sucrose also we form that glycosidic linkage, yes or no? So here also N glycosidic bond will be formed. N glycosidic bond will be formed. Clear? So let me form the bond. Arey, kahan gaya structure? Where is the structure, guys? See here. So this carbon, from this we are going to remove the OH. From this we are going to remove the OH, and what we are going to do? We are going to remove this H. We are going to remove this H. Where it's gone? 1 second, bachcho. This H. Okay, this H, and we are going to form a bond. And we are going to form a bond here. 1 second, 1 second. So I'll not making it whole. I'll just make this structure. In this way, it is going to form the bond. Yes or no? Yes or no? Clear? Okay. So this bond is known as what? N-glycosidic linkage. N-glycosidic linkage. And which nitrogen participates in this glycosidic linkage? Which is 9th nitrogen. Which is 9th nitrogen. Clear to everyone? Which is 9th nitrogen. Is it clear? Is it clear, guys? Okay, chalo. Now what? Now we have the DNA. Okay, let's talk about the DNA, people. Let's talk about the DNA. Any doubts? Ask me. Any doubts? Ask me. Bala and Shivesh. Okay, great. Chalo. Let's talk about the DNA. Acha, one more thing. Adenine plus, I'll give you the question. 1 second. Before moving, I'll give you the question. If I have adenine, okay? If I have adenine plus ribose, then what will happen? Will you form adenosine or will you form what? In case of adenine, but in case of pyrimidines, yes, exactly. Adenine plus ribose, what will be? Will it be called as, what it will be called as? Adenine plus ribose. We are going to call it as adenosine. And if we have adenine plus deoxyribose, then what? Sir, it will be deoxyadenose. Deoxyadenose. Yes or no? Yes or no, people? Let me know. Yes? Chalo. Great. So you will do uracil plus ribose. You will be able to write uridine. Cytosine plus ribose. You will be able to write cytidine. Yes or no? Guanosine plus phosphate. It will be what? Guanylate acid, GMP, AMP. Yes or no? Should I write all the things? No, not required. Okay, chalo, great. Now let's move on with the core of the chapter now. Okay, let's move on with the core of the chapter, which is the structure of DNA. What's the full form of DNA? Shrishai got scammed. Actually, Shrishai, what happened? There was confusion between me and my team regarding the timings. So that's why it happened. I am so sorry, Shrishai. It's literally my mistake. They have scheduled it for 7:35, but I thought I take class at 6:30 usually, so I started at 6:30. I am so sorry. Chalo, let's talk about the DNA. Let's talk about the... Why are you attending these classes already in AIMS? Because I am a very good teacher. That's why AIMS students are also coming. It's not like that, Manjit. He's also preparing for NEET. Okay, chalo. Arey, wapas aao, come on, let's get back here. So DNA is what? DNA is acidic or basic, sir? DNA is acidic. Yes or no? DNA is acidic. Okay, now it was identified by Mishra. Which Mishra? Remembered Bala Shivesh, that Mishra only, which went to the park and got hurt and got pus. Yes or no? That Mishra only. So DNA was first identified by the same person, which is F. Mishra, okay, which got hurt and went to the park and then observed the nucleic acid in the pus cell. Yes or no? Hi CRC Yadav. Clear? So DNA is acidic and it is first identified by F. Mishra. But the term DNA, see, F. Mishra was a person who was not very much intelligent. Okay, he was not very much intelligent. He used to do the discovery and he used to say other persons to give the term, right? So Altman gave the term nucleic acid there, and here DNA the term was given by Jackaris. DNA the term was given by Jackaris. Clear? Any doubts? Any doubts anywhere? So DNA is an acidic molecule. DNA is what? DNA is the acidic molecule first identified by F. Mishra, and then what? Then the term given by was Jackaris. Who gave the term? The term DNA was given by the Jackaris. Clear to everyone? Any doubts? Chalo, great. Now let's move on. It's made up of what? It's made up of what? So I'll give you a question. DNA consists of number one, nitrogen bases, ribose sugar, third will be both, and fourth will be none. Give me the answer fast. That's a very easy peasy question. A very easy peasy question. I just want to check your presence of mind. A very easy peasy question, everyone. Yes or no? A very easy peasy question. Tell me the answer. Manjita, Shivesh, Bala. Option, Sri Shai, option. Give me the option. 1. Why not both? Why not both, guys? Why not both? Shivesh, why not both? And now someone will be there who will be typing both as the answer. See, Shivesh did it. Proud of you, Shivesh. Bhai, it's wrong. It's totally wrong. I would have written deoxyribose, na? Second option, both will be then when I would have written deoxyribose. In DNA, there is no deoxyribose. There is no ribose sugar. It's deoxyribose sugar. Yes or no? Yes. So DNA consists of what? DNA consists exactly Sri Shai. DNA consists of DNA consists of nitrogen bases and deoxyribose sugar. Yes or no? Okay. Now, what's the charge of DNA? Nitrogen bases is there plus deoxyribose sugar plus phosphate. And what's the charge of DNA? Is it positive or negatively charged? So, sir, the DNA is negatively charged. The DNA is exactly negatively charged. And what's the charge on histone protein, guys? I know I have not told histone protein right now, but you might have studied what will be the charge of histone protein. Histone protein somewhat makes DNA stable. So, what should be the charge of histone protein? What should be the charge of histone protein? Yes or no? Positive, exactly. That's only the negative and positive will make each other comfortable. Yes or no? Yes. So in a marriage, if both the person are very arrogant and both the person wants to fight, so they will soon have divorce. Yes or no? So whenever you guys marry, what you do is that search the person having the opposite characters. Yes or no? So that if you fight also, that person will say, I won't fight. I don't like fighting. Yes or no? Okay. Chalo. Now let's get back here. In DNA, what kind of nitrogen bases are present? In DNA, what kind of nitrogen bases are present? Tell me. In DNA, what kind of nitrogen bases are present? ATGC. What? Adenine, thymine, guanosine and cytosine. Yes or no? Adenine. Okay, great. Adenine, thymine, guanosine and cytosine. Yes or no? Yes or no? Okay, great. Uracil is absent. Exactly, uracil is absent. Now guys, tell me one thing. Who discovered the structure of DNA? Like there is lots and lots of scientists, yes or no? But there were four scientists mainly involved. There were four scientists who were mainly involved. One is Wilkins and Franklin. One is Wilkins and Franklin. This so you remember it. This scientist so you remember it. Wilkins and Franklin. It comes in NEET. So Wilkins and Franklin don't guide us about the marriage. Think yourself first. Rajdeep. Don't worry guys. I will search a good person for me, right? Chalo. Watson and Crick. Exactly. Chalo. So Wilkins and Franklin is what? Wilkins and Franklin is basically what? They went and they took out the X-ray of DNA molecule. What they did? They took out the X-ray of what Wilkins and Franklin took out the X-ray of DNA molecule. What it did? They did X-ray crystallography. X-ray crystallography, okay? What they did? X-ray crystallography. Wilkins and Franklin X-ray. You will search a boy, I guess. Why? X-ray crystallography. X-ray crystallography is what? X-ray crystallography is of DNA. Yes or no? Wilkins and Franklin gave what? My God, what I am saying. Wilkins and Franklin did what? Did X-ray crystallography of DNA. Okay. Now, with the help of what? With the help of another two-person scientists, which is Watson and Crick. What they did? They gave the structure of DNA molecule. They gave the structure of DNA molecule. Okay, stop planning about my wedding. I'll call you each and everyone. I'll invite you in my wedding party. Okay. पहले पढ़ लेते हैं, right? चलो. So Watson and Crick. Next we have Watson and Crick. So Watson and Crick what? Watson and Crick studied the structure of DNA. Watson and Crick studied the structure of DNA with the help of the results given by the X-ray crystallography, DNA X-ray crystallography, which was given by Wilkins and Franklin. Okay, which was given by Wilkins and Franklin. So Watson and Crick and Wilkins and Franklin, they did what? They gave the structure of DNA. Yes or no? They gave the structure of DNA. Yes. And after this what? After they gave the structure of DNA, Watson and Crick received what? Watson, Crick and Wilkins received the Nobel Prize. Watson, Crick and Wilkins received the Nobel Prize. Is it clear? Is it clear to everyone? Watson, Crick and Wilkins received the Nobel Prize. Guys, Franklin didn't receive the Nobel Prize. Why? Anyone in the comment section? Guys? Anyone in the comment section? Why did Franklin didn't got the Nobel Prize? Because he was dead. Okay? Clear? Yes or no? No, because he was dead. Clear? Okay. Great. Now, Franklin died. Exactly. Franklin died. Franklin died. Clear? Any doubts anywhere? Any doubts anywhere, guys? Any doubts anywhere? Any doubts anywhere, people? Tell me. Any doubts anywhere, people? No doubts? Okay. So what we studied today, we studied about the structure of the we studied the nitrogen bases, we studied the structure of the sugar, we studied the phosphate, we studied the phosphate bond also, and then after that we studied the DNA, like it's the properties of DNA, which is acidic, which is negatively charged, first identified by F measure DNA and all those things, and then we saw the who gave the structure of DNA. Clear? So in the next class, what we are going to do is that in the next class, we will be starting with the structure of DNA. In the next class, we will be starting with the structure of DNA. Clear to everyone? Okay. Yes, we will be starting with the structure of DNA, Chargaff's rule, configuration of DNA molecule, all these things we will be doing with the next class. Okay. So don't forget to subscribe to this channel and join in the next class. Till then, bye bye everyone. See you in the next class.